The effects of the different concentrations of aspirin solution in the seed germination of a tomato plant. The Philippines, being known for rich agricultural land, is full of farms and factories meant to operate for the agricultural sector of the country. This sector, according to Bacongas, contributes drastically to the country's gross national product, making it a crucial concern for the Filipinos. Nowadays, farmers are facing numerous problems regarding their crops, pests, diseases, and the weather are some, to name a few. These heavily affect the growth of their crops, reducing outputs. With these in mind, the researchers had set their sights on aspirin. Aspirin contains salicylic acid, which is proven to help with the seed germination of a plant. Since the given time frame is suboptimal, the researchers settled with a tomato as the plant to be worked on. This is due to the fact that tomatoes germinate seeds quickly. The plant itself is not the only present variable in this study. Other components such as the other parts of the tomato plant, the soil, the environment, and the researchers themselves are exposed to the aspirin solution. Another thing to consider is whether or not the fruit is safe to consume after developing with the help of the aspirin solution, its physical changes, and the differences in its physique as well. These variables tend to conclude both positive and negative effects. Take note, the effects varies on the volume, frequency, environmental factors, and the test subject used in each experiment. Before we dive into the study more, allow me to discuss some of the related literature and studies essential to the framework of the study. It's a synthesis of literature and researches that supports the use of aspirin on seed germination. The goal of this study is to determine how well aspirin works with accelerating the process of seed germination. But first, what exactly is seed germination? Seed germination refers to the physiological process culminating in the emergence of the embryo from its enclosing coverings. The absorption of water by the seed activates metabolic processes that lead to the expansion of embryo and penetration of organ through surrounding tissues. The transcriptions of germination-related genes and their translation into proteins begins within the first few hours following hydration. Expansion of the embryonic tissues is opposed by the restraint of tissues enclosing them. An increase in embryo growth potential and for a decrease in the strength of the covering tissues allow germination to be completed. Germination begins with a water uptake by the seed and ends with the emergence of the embryonic axis. Take note, it does not include seedling growth. This happens after germination is completed. Since the tomato plant will be the test subject, here are the necessary conditions for the seed germination of the said plant. The conditions needed are preconditioning, moisture, oxygen, and warmth. Preconditioning refers to the specialized process done to the seed before they germinate. Some examples of preconditioning are alternate freezing and the use of red light and infrared light. They are known to produce bioactive chemicals called gibberellins, which promote seed germination. Moisture is needed sometimes because they stay dormant as long as they are dry. Like humans, oxygen is needed by dormant seed to breathe or respire. Warmth affects the seed germination of tomato plants. They best germinate between 17 Celsius and 20 Celsius. A tomato seed, as stated by Collins L. Morrow, can be considered to have successfully germinated when two distinctly separate cotyledons, or embryonic leaves, can be seen. Another essential subject for this study is aspirin. Why did the researchers use aspirin as the agent for this study? Well, aspirin, also known as acetylsalicylic acid, has analgesic, antipyretic medicine made by salicylic acid. Interacting with acetic anhydride, it is a white crystalline powder that is odorless and stable in dry air. As stated by Waller et al., It's mainly used as medicine, often used to alleviate pain and swelling. Moreover, plants with stress produce minute amounts of salicylic acid on their own. This small amounts aids the plant to cope when under insect attacks, under dry, or even when experiencing a disease. Similarly to humans, the component helps boost the plant's immune system.